Thanks, and thanks everyone for tuning in. I'm Fernando, and here I'm going to tell you how we discover a transcription factor in Archaea that we think may tell us something about the early evolution of eukaryotic transcription factors. I'm currently a postdoc at Dipti Nayak's lab in UC Berkeley, so I would like to start thanking this amazing group of people for making this work possible. I also love presenting this work because it gives me the opportunity to introduce you some of our non-official members of the lab, these little squirrels that go visit us every day, and for a good reason, because we feed them. This is a video of a squirrel that's been trained to ring a bell from the outside of this window, so we know that they're there, and then we can feed them. But by doing so, I realized something unusual. Turns out that these squirrels are very picky. They will rather almonds over everything else, um, which is analogous to what we do every day in the lab when we need to feed our microorganisms that we culture. Some of them turn out to be very picky, and that's the story that I'm going to tell you today. We study the physiology of archaea. You might have heard them as this third domain of life. They are microorganisms that morphologically are very similar to bacteria, but rather genetically, they're more closely related to eukaryotes. They're more similar to us. So they are a good opportunity to study the early evolution of eukaryotes. In the lab, we use the model organism Methanosarcina acetivorans, which as a metabolism uses methanogenesis as a form of living. For methanogenesis, they can use different compounds from the environment. They eat different compounds, such as trimethylamine or TMA, which when it's available, it enters the cell and the cell expresses or produces a specific enzyme, this green MT1 enzyme, that is able to eat TMA. It's able to take away one of the methyl groups of this TMA molecule represented by the blue circle and transfers it to a different carrier protein that then is transferred to a carrier molecule to enter the core methanogenesis pathway, which then these organisms use to produce methane, which they couple with energy production. If trimethylamine is not available in the environment, but rather other compounds that they can use is available, such as methanol, what they do is that they completely shut down the pathway for trimethylamine and instead they produce or express a different enzyme, this orange empty one, that now it's able specifically to take the methyl group from methanol in red into the core methanogenesis pathway. How these organisms are able to sense and determine whether they have available methanol or TMA in the environment is something that we don't know. And we also don't know how this organism is able to express the correct enzyme based on these environmental cues. This is important because every cell needs to know what they have available in the environment. So to study that, we did a simple laboratory evolution of this organism where we culture them anaerobically in these test tubes, for example, with one particular substrate like methanol. But then when these are done, then we transfer an aliquot of this culture to a medium that has now TMA. And when these are done, we transfer them back to cultures that have methanol. We did so over and over again, alternating the carbon source for 50 times. And then at the end of this sequence, we wanted to ask a few simple questions. First, do the cells adapt? And the answer is yes. Turns out that the cells are able to switch faster between substrates at the end of the experiment. Second, are there genomic changes that explain this adaptation? And the answer is yes. We found mutations in one single gene, a gene that hasn't been studied, that is only a number in the genome of this organism, which is MA2561. And lastly, we wanted to know how is this adaptation happening? We wanted to know the mechanism for adaptation, and we found out that the protein produced by this gene, 2561, is actually a transcriptional repressor of growth on TMA. So to understand the molecular basis of this repression, we can visualize the structure of the protein that is produced by this gene, 2561. This is a molecular model of the structure. We found out that this protein only has two domains, this GAF domain and PAS domain, both associated with proteins that are able to sense the environment. They bind small molecules. So it makes sense that if TMA is available in the environment, maybe this protein can bind TMA. And we actually tested that in the lab, and indeed we found that the protein is able to bind TMA. So for our model now, we know that the protein is a sensor of environment of TMA in the environment. But the most intriguing question is to determine how this protein is able to transduce this information to express the appropriate, the proper genes for the pathway of trimethylamine. This is relevant because this protein only has these two domains. It doesn't have any discernible DNA binding domain. So for a long time, we thought that the protein may interact with other proteins to do this job, but turns out that that wasn't the case. Instead, one of the things that we found is that the protein forms dimers. And so now if we model the structure of a dimer for the protein, some of the things that caught my attention is this area of the protein here, these little legs that look very similar to transcription factors that are only known in eukaryotes, such as, for example, this basic leucine zipper, which uses similar legs or this coil domain that grips DNA. Another feature of this transcription factor is that they are positively charged. So if we visualize the surface electrostatic potential of this protein, in blue, we see the amino acids that are positive that counteract the negative charge of DNA. And if we visualize our protein now by surface electrostatic potential, we found out that in this leg, we also have positively charged amino acid. So we thought maybe, just maybe, despite not having any discernible DNA binding domain, this protein is able to bind DNA through a mechanism that is more common in eukaryotes, and it's not known in prokaryotes. And we tested that by doing an electrophoretic mobility shift assay, or EMSA, where we took the promoter of a gene that we know that is affected by this protein. And the DNA sequence of this promoter, we tag it with a fluorescent probe so we can visualize that on a gel. This is how a 100 base pair probe looks on a gel if we don't add the protein. But if we start adding the protein to these gels, when we run the protein and we add more and more protein, we found a shift of this band towards lower electrophoretic mobilities that represent the DNA protein complex, meaning that the protein indeed is able to bind DNA, likely using this non-characterized domain for DNA binding. So now we know that the protein is able to bind trimethylamine, but when trimethylamine is not available in the environment, instead it binds DNA and it represses the genes that are essential for growing on TMA, which explains what we saw initially in the experimental evolution. For this reason, we call this protein AMCR, or Archaeal Metabolite Sensing Zipper-like Regulator. And we think that these regulators are very important because, as far as we know, this is the only example of a prokaryotic transcription factor that looks like a eukaryotic transcription factor, which tell us something about the early evolutionary history of eukaryotes, which fascinates me. What I have here at the bottom is a geological time scale with the different eons. Here's where Earth was formed. This is where we're at. One of the biggest questions in biology is when and how life emerged, followed by when and how eukaryotes emerged on Earth. And we have a range of time of over 1 billion years based on the early fossil record of eukaryotes. We have some very good ideas about how this transition happened, but what we don't know very well is how complex regulatory networks in eukaryotes that allow the diversity and complexity of eukaryotes, how we don't know how those evolve. And I think that maybe we have the key in these archaeal proteins, these archaeal transcription factors, that were a prokaryotic invention somewhere before the emergence of eukaryotes, and then they were inherited into to eukaryotes. So that's why I think studying these proteins is very important to understand the early evolution of eukaryotic transcription factors, making them essentially living fossils of early transcription factors in eukaryotes. We already studied some of these proteins, some other homologs from related organisms. All of them are sensors for TMA. But we believe that other proteins in Archaea hold the key to understand these transcription factors because they may have diverse different functions. The problem is that we cannot identify these proteins by bioinformatic methods. So we need to do other functional analysis to understand how these proteins operate, what is the mechanism of DNA binding. I would also like to complement all of this molecular information with the early fossil record of eukaryotes, taking advantage of my prior knowledge and background, studying the fossil record to have a more holistic understanding of how early life evolved on Earth. This work is available on bioRxiv for now, if you want to know more about it. And if you have any questions, you can send me an email. I would also like to thank my lab again and the different funding institutions for making this work possible. Thank you so much.